Yeah, hello everyone. Welcome to the post-lunch first session. You see your bright-eyed and bushy-tailed after a great meal of good food. This next session is restoring oyster populations, and it's really a timely thing. Oyster populations in the Great Bay Estuary have been really affected by a lot of different ways of changing their population, including a lot of harvesting through the decades, actually through the centuries pretty much. We got hit with oyster diseases back in the mid-90s that really decimated the population. So the population of oysters in the estuary has really gone through a lot of changes, especially over the last 20 years or so. So this session is really timely. We're going to be we're going to have 3 talks. Kelsey's going to talk about supporting oyster aquaculture and restoration. Jannine is going to talk about insights from a citizen science program to support oyster survival. And Katie's going to talk about Seascape, genomics, and common garden experiments. So a wide array of approaches to really trying to address this really key part of our ecosystem. Kelsey?
All right, good afternoon, everyone. So I am Kelsey Meyer-Rust. I am the Coastal Conservation Coordinator at the Nature Conservancy here in New Hampshire. And today I'll be talking about some of our oyster restoration work, specifically a program called SOAR, or Supporting Oyster Aquaculture and Restoration. And so Steve kind of briefed on this already, but our Great Bay wild oysters have greatly declined. They're about 90% less than what they used to be. And this is due to, a wide variety of reasons, but some 4 main ones are disease, such as MSX and dermo. And with our waters getting warmer, these diseases are going to become more prevalent, so more of an issue for our oysters. Sedimentation, this makes it a lot harder for oysters to actually settle on favorable substrate. Historical overharvesting, and then invasive species such as green crabs and oyster drills, which can be pretty big predators on our oysters. And so, why oyster restoration? So oysters are like these little powerhouses. They have a slew of economic and ecological services. And so we want to restore our oysters so that we can get all these great services that they provide us. They help a lot with coastal economies. So we have, New Hampshire oyster industry here. So we want to help support that. They also help a lot with water quality, being filter feeders. An oyster can filter out about 50 gallons a day. They're also great habitats for other animals and plants. And they've actually been shown to increase fish production. And then they also help with shoreline stabilization as well. So these are all reasons as to why we want to help restore our oyster reefs and Great Bay Estuary. And so this program SOAR, it has multiple components, but due to time today, I'll just be focusing on our purchase program. So SOAR is kind of broken up into 2 different phases. So phase 1 has been completed already. So just to give a little bit of background on that, this actually started out as a COVID-19 relief program, kind of like a win-win situation. Our oyster farmers were having a hard time selling a lot of their oysters. Restaurants were closed. People weren't buying oysters. And we also wanted to partner with farmers to get these single live oysters out on these restoration sites. So they were able to get some supplemental income and we were able to get these oysters. So definitely a win-win. And between 2020 and 2021, we deployed over 650,000 of these adult oysters, about onto 2 acres in Great Bay. And we partnered with 12 farms in New Hampshire and then 16 farms in Maine. And we were actually able to see some of the highest recruitment on nearby native reefs in about 10 years. And so now we're kind of on to SOAR phase 2. And so this first line, this is everything we were able to accomplish in SOAR phase 1. And with SOAR phase 2, these are kind of our target outcomes. But in general, we're trying to combine, like between SOAR phase 1 and SOAR phase 2, we want to deploy about 6 million oysters onto about 70 acres of reefs and engage with 175 oyster growers. So these are kind of the things we're trying to target and some of the things we've already been able to accomplish. So for SOAR phase 2, for deployment, we had about 150 cubic yards of mulch, which is just made up of clam and oyster shell. We partnered with our New Hampshire oyster farmers to deploy spat on shell, and spat is just like a baby oyster, and I'll have a picture of that at the end. And then mixed with some large single oysters as well. We're looking at monitoring recruitment, survival, and growth to see how these restoration sites are doing. And we're also quantifying fish production, denitrification, and conducting shoreline profiling at these restoration sites as well. And just to kind of give a brief overview of our aquaculture industry here in New Hampshire. So again, we partner with our farmers. It's definitely a newer industry, but it has been quickly growing over the past few years. So currently we have about 12 active farming businesses, just over 100 acres of that. And most of our farmers are in the Great Bay watershed. In 2024, they harvested 1.2 million oysters and provided about $4.5 million in ecosystem services. And just want to give a shout out to our farmers that we're able to partner with this great program. And so on the left here, we have a map of Great Bay Estuary. These green dots, these were restoration sites that started in 2000 and ended in 2022. But us, the Nature Conservancy, we started doing oyster restoration in 2009. So we've been kind of doing this for about 15 years. But other folks like at UNH have been doing this since 2000. You can see that the green dots, they're all kind of adjacent to like these orange splotches. Those are our natural oyster reefs. And these two stars at the very bottom at Nanny Island and at Squamscott, these are our current oyster restoration sites. Nanny Island was for SOAR phase 1, and now Squamscott is for SOAR phase 2. And on the right here, we have a map of the Squamscott River. We have about 2.5 acres permitted for oyster restoration. And that upper white box, that's kind of the actual restoration site. So that's where we need to put the mulch, the spat on shell, and those live oysters as well. And it's kind of adjacent to that wild Squamscott site. So this here, this is for Nanny Island. So we were able to monitor Nanny Island through different things like tonging, video, dive surveys. And so we have a bunch of green, yellow, red, and blue dots, all representing different years. I mostly just wanted to point out those blue dots. So that's from surveys taken in 2024. And you can see those dots are a lot larger and there's more of them. So this is kind of showing proof of concept. With this restoration work, oysters are surviving and we are getting recruitment. So that's super encouraging for SOAR phase 2. Because SOAR phase 1, we are able to see those oysters surviving, recruiting. So this is working. And so how can we measure the success of the SOAR program? So we're working with a postdoc researcher at Northeastern University. And these are some of the key questions she's hoping to answer with our data. So these questions she's hoping to answer over the next year, as if we're in our last year of implementation right now. But I'm just gonna get some quick data on the survival of seeded oysters by size class at Squamscott. So this is kind of the history of Squamscott over the past few decades. So this is at the wild reef site, not the restoration site. So this is a scatter plot she was able to make using data that Fish and Game collected doing dive surveys. So I just mostly wanted to point it out that, so the red dots, those are recruits, so anything less than 20 millimeters, and the blue dots are adults, so anything about larger than 61 millimeters. And we can see kind of a correlation with the increase of adults with the increase of juveniles. And also there's no real population crash here, which is really great. So making Squamscott kind of a good site to have an adjacent restoration site too. And then this is the actual SOAR restoration site, so that area adjacent to the wild reefs. So this was data collected by PrEP in 2024, doing kind of quadrats, doing dive surveys. And so we have single oysters and spat on shell. The single oysters were put down in 2024, and then the spat on shell is kind of a mix of 2023 and 2024. So you can see in the middle, we have a lot of these large blue lines for the spat on shell. These are spat on shell that were put down in 2023. So we can see that they are surviving and growing, which is really great. So that definitely gives us some encouragement for those dive surveys that will then be taken this year to look at the stuff that we put down in 2024. And then we also put spat on shell and singles this year as well, and we're still in the process of doing that. So for the future of SOAR, kind of over the next year, we'll be putting together a best management practices document. We're doing some more deployments right now and also just monitoring activities over the next year. We're also hoping to answer all those research questions that the postdoc at Northeastern will be doing, and then also hoping to get some publications out of this as well. And then just wanted to point out this image right here. This was taken just a few weeks ago. So this is kind of what the spat on shell will look like. This is kind of like the golden standard of spat on shell. They're growing, the oysters are kind of clumping together. So this is the type of stuff we want to put on those restoration reefs. So hoping that kind of clumping will attract more oysters. So we had a really good successful year of spat on shell this year. So we're pretty excited about that. And yeah, thank you.
Thank you, Kelsey. Our next speaker is Jannine Chamorro. And she'll be talking about insights from the citizen science programs for oyster survival and growth.
Hello, everyone. So I'm Jannine, and I am a postdoc in the Quantitative Marine Ecology Lab at UNH. And today I will be talking to you about a citizen science program that the TNC has run for almost 20 years now, and what some insights we can take from this program to help support oyster survival and growth. All right, so for almost 20 years now, TNC has run this community-based restoration program, and the way it works is that every summer they give volunteers around the Great Bay Estuary cages containing shells that have spat on them. And so what the volunteers do is that they monitor these cages and they maintain them. And what part of the monitoring efforts include measuring spat survival and also growth. And generally this runs from July to September every summer. And the 20 years that this has gone on there's been lots of efforts, and it has contributed to almost or greater than 300,000 live oysters in the bay, which can help restore vital ecosystem services. So over the years, what they've noticed is that across the Great Bay Estuary, there's some differences in size and survival. So looking at these two images right here, on the right side, we have a estuary site, and on the left side, we have a coastal site. And you can see that the spat in the estuary generally a lot bigger than the spat on the coastal site. So the data that is collected again is spat survival and growth. And because we have so many volunteers that participate in the program, there's comprehensive spatial and temporal coverage. So with this data, we also have environmental data that's been collected by NERS and PrEP, which includes temperature, dissolved oxygen, salinity, and chlorophyll. And so what we're interested in doing is pairing environmental data with oyster survival and growth data to help us determine what environmental conditions specifically contribute to oyster survival and growth. Yeah, specifically what water quality conditions optimize spat survival and growth. And sorry for the order of this. The sites that I have specifically focused on are the Squamstruck River, the Lamprey River, the Oyster River, and the Great Bay, just because this is where we have these long-term water quality stations. And here I'm gonna be talking to you about the growth data, just because that's where we've seen some of the biggest differences. If you look at this density plot here, you'll see that generally the sites are pretty similar. In terms of growth, maybe the Squamstruck River the growth is a little bit smaller than the other three. But when you break it down between the different years, you can see that here we have 3 consecutive years, 2016 to 2018, with location on the x-axis and growth on the y. But there actually is some variation between sites across years. When I've incorporated some of the environmental data and some of these analysis, generally we see that temperature has the greatest effect on spat, where as temperature increases, growth seems to increase as well. Interestingly, the water quality parameters that I've looked at don't seem to impact survival as much. And so some next steps for this data analysis are to include whether these spat on shell are intertidal versus subtidal, because that will have a big impact on survival and growth. If you're intertidal, you're in harsher conditions, but also you don't have access to food as often as if you're subtidal. Also including physiologically relevant metrics. So for a lot of the analysis that I've been working on right now, it's in looking at like mean salinity, like daily mean salinity or over the season, but it's important to look at like, okay, how much time they spent at like hypoxic conditions would be interesting to know. And then also any other metrics that you all would be interested in looking at in terms of how they might affect early stage oysters. Thank you.
Thanks, Jannine. Moving right along. We're gonna hear about Seascape genomics and common garden experiments from Katie from Northeastern University.
Thanks so much. Okay, so I'm going to talk today about an experiment we've been doing down in the Chesapeake Bay, but we are using Great Bay oysters in that experiment. And to start off, we could think about how we choose individuals for a restoration project. And there's 3 kind of frameworks we could use to choose those individuals. If we think that local conditions select for a genetic type, like they're locally adapted to those local conditions, we would just choose local, and that framework we call local is best. But we could say maybe the environment's changing, climate's changing, and there's some maladaptation to local environments. And in that case, we might want to pick individuals from further south for a restoration experiment. In this case, we would say we want to minimize environmental distance. But we might not know what the environment is. We might not know what the genetics is. And so it might be better to just maximize intraspecific diversity and use a diversity of genotypes and hope that selection can kind of sort out which are going to be the best ones. So to compare these frameworks, we would have to do some kind of experiment and where we have what I'm going to call monocultures, where there's oysters collected from a single population. You'll see some slight color variation here representing some genetic diversity within that population. We compare them to non-local genotypes from the south, as well as non-local genotypes, from the north, which is colder, from the south, which is warmer. And we create some polycultures in terms of a mixture of genotypes. And we can also think of polycultures in terms of crossing different genotypes from different populations and creating new genetic combinations for restoration. So to do this, you also need environment of origin data for each genotype, environmental data for the common garden sites, genetic data for each group, amazing collaborators to help you pull this off, an amazing team of students to do all the work, and funding. And luckily, we were able to pull all that together. My collaborator, Jess Small at the Virginia Institute of Marine Science, runs a research hatchery down there. And the work I'm going to be showing you today is done by graduate students and undergrads in my lab with funding from the National Science Foundation. So in our common garden experiment, we raised oysters from, that were created from adults collected from Texas, Louisiana, Florida, 3 local sites in the Chesapeake Bay. Within those, 2 of the populations were selection lines. One was selected to lower salinity, one was selected for a higher salinity site. And then W5 New Hampshire is our Great Bay oysters, and we also have oysters from the Daramescotta in Maine. And we had 2 common garden sites, a low salinity and low disease site at Louisetta. So L means low salinity, lower disease, and the York River was a higher salinity and higher disease. We also created 2 polyculture treatments, one where we mix seed together from these different oysters, and another one where we actually crossed gametes from the oysters and created hybrids and mixed them together. So this was quite a lot of work. It looks like maybe my video didn't transfer here. I was going to show you a video of the spawning. But basically, like we had to like keep all of these oysters in containment when we brought them in, and until we could spawn their seed, and then we could put their seed out in the field. So it was a big team effort. We did all of this on one day, very long day in May in 2023. And then we put them in these bags out in the field. This is another video that's not playing, but we monitored the survival and growth of oysters in these bags through time. Over that period, we've counted oysters as alive or dead 155,000 times. We've tagged over 2,000 oysters and measured them almost 8,000 times. We collected a ton for in-depth phenotyping, looking at condition. We collected oysters to monitor disease pressure. And we managed all of this data in an app. So I'm really into app building if anyone wants to chat about that. So in these 2 common garden sites, this is the dermoprevalence disease. So basically in Louisetta, disease reached a prevalence of 50 to 60%. That's the lower salinity site. In the York River, disease pressure was very high. Almost all oysters were exposed to the disease. And this is salinity data. So the Louisetta salinity is typically below 15 parts per million or parts per thousand. In the York River, it's typically above that. So here is the data for the 2 sites across those different groups. And this data was analyzed by my graduate student and undergrads. And just to break it down for you, we're going to start with that local is best framework. So we did find in the Louisetta site that the local selection line Lola performed about the same as sites from the Gulf of Mexico. So sites from a lot farther south in Florida and the Gulf perform the same. So not evidence for local is best. In the York River, with these darker boxes now, I'm showing you the sites that did the best. And again, we find the selection line that has been selected for the conditions there did about the same as sites from another site in the Chesapeake or sites further south in Texas and Florida. We can now ask, what about maximizing intraspecific diversity? Do these polycultures do any better than the other groups that we had? And the answer is they performed about intermediately. So the polycultures did not outperform any of the monoculture groups. So next we can ask, well, what about minimizing environmental distance? Is there some environmental matching that's helping to explain these patterns? So we have salinity data and temperature data from all of the sites. And I think the important thing to notice here, is that New Hampshire and Maine do not reach the maximum temperatures that are being reached in the Chesapeake Bay in this experiment. But we do have sites from a variety of different salinities to compare to the salinity and temperature in the field. Yeah, so those Northeast oysters experience lower temperatures. And so the environmental distance hypotheses we would predict that survival would increase with genetic diversity and decrease with environmental distance. So we included both of these metrics. Now we're just looking at those monoculture treatments, which do come from populations with different levels of genetic diversity. So the slopes between different variables and survival are shown on the x-axis. And as you can see, we see positive slopes with allelic richness and heterozygosity, which are measures of genetic diversity. But we see positive slope with environmental distance, and that's not what we were expecting. So instead of looking at environmental distance, which is like a multivariate measure, we could ask, well, what about environmental values? Is there something about salinity at the environment of origin or temperature that correlate with survival? And we expected that the oysters from saltier sites would do better in the York River, and oysters from lower salinity sites would do better in Louisetta. So we were expecting this interaction shown in the lower left. So again, here on these graphs on the right, I'm showing you the slope on the scale data. And we do see that genetic diversity again increases with survival. I forgot to mention sites with or populations with higher genetic diversity also came from warmer temperatures. So we have some confounding there. And in the York River, we saw this positive relationship between salinity and survival, but we didn't see that in Louisetta. Instead, in Louisetta, we saw the same pattern. Even though it's a lower salinity site, oysters that came from higher salinity places had higher survival. So to summarize, what kind of support do we see for these three frameworks? Well, we didn't find strong support for the local is best hypothesis. We found that oysters from quite distant sites further south did just as well as the local selection lines and wild population. For maximize intraspecific diversity, we have mixed support. When we created those polycultures, they really didn't do better, I think, because there was just too much of a mix of genetic diversity in them. But oysters that came from populations with higher genetic diversity tended to have higher survival. And with regard to minimizing environmental distance, a simple multivariate measure of distance didn't seem to capture the environment in the right way. But groups that came from southern parents that were warmer and had higher salinity had higher survival at both sites. And I think, you know, there's a relationship between disease pressure and salinity and temperature in which these oysters would have also had higher history of disease pressure. And I think that that's probably explaining some of the patterns that we're seeing. So with that, I would like to thank you. I do have a lot more genetic data of oysters across the entire Atlantic seaboard, if anyone's interested in seeing that, and I'd be happy to discuss this some more. Thanks.
Right. Thanks, Katie. Do we have time for questions?
Yeah, maybe like 8 minutes.
8 minutes. Who's got a burning question?
10.
10. What?
Okay, thank you very much. Just a question regarding the hybrids and the book. So you said that for the last experiment, the mixture didn't work well because there was high genetic diversity. And also the other thing is that why the hybrids you didn't get the expected hybrid pattern.
So we just, so in the polycultures, they would have consisted of the genetics of individuals from all the way from Texas to Maine, right? And so the individuals, the northern populations from the Great Bay and Maine did the worst in the Chesapeake. They had the lowest survival. So when you create polycultures with populations that have low survival and some populations that have high survival, you end up with polycultures with intermediate survival. And I think that's what we saw there. I was surprised that there was no real statistical difference between the mixture of seed versus the crossing of gametes to create hybrids in the pattern. I would have expected some differences there, but we didn't see it in the survival metric.
While you're thinking of questions, I was wondering, for Jannine and Kelsey, we've been sampling oysters in the Oyster River, a little further up in the older restoration site. We didn't get any this last time. This is the first time you get one that was about this big, so we can analyze it, but we've got one cohort, and we got no oysters, so. Wondering if you guys have any insight into what's going on in that Oyster River.
So at the Oyster River, we do actually have like a oyster community garden there where, we have this like wooden structure set up that, actually a Boy Scout group made for us, which is cool. And we have these, oyster cages hanging from there that have the spat on shell, and we have volunteers to go check out on those cages for us, a couple times throughout the season. And I will say, that site definitely did not do as well as some of the others. I'm not fully sure why that site doesn't do as well, but we saw something similar where the spat weren't growing or surviving that well at that site, but I think that's kind of the questions Jannine's like trying to answer.
Yeah, I went out there every week this summer, and it just, I'm also new to the system, but it was very muddy, and also when I would measure water quality parameters, it seemed like DO was all sometimes really low there, and the water levels were just really low in that Oyster River City.
Okay, thank you. Jeff?
Okay, so curious about eco-evo feedbacks in this, the common garden experiment. Would you expect the results to be similar on natural reefs, as opposed to, I suppose the common garden is a little bit more like it would be in an aquaculture situation?
Yeah, I meant to mention that we raised these oysters in bags so we could monitor their survival through time, so it was more of a aquaculture context than a restoration context. With the restoration question, I think, you know, there might be more predation on spat than we would have had it naturally in our experiment, so I think that's a question for restoration. But there's like the population level differences are really huge. I mean, I think it just illustrates that there is a really important genetic component to consider and test for in these projects.
Yes.
This is Mark. Awesome, so much to think about. In terms of the restoration, do you think that there's a limitation in terms of space for natural recruitment to be happening, like structure or substrate?
That's a good question. I'm not sure if that we're permitted to a specific acreage, so like what we put down is in a certain area. So like at a certain point, we'll be limited how much substrate we can add. So you know, that natural recruitment will hopefully go there. Yeah, like we're permitted to a specific site, so we can't like go outside those perimeters. So I guess like in a way that could technically limit how much recruitment we get with the substrate.
But in the areas outside of where you're adding substrate, do you think that there are natural limitations in terms of how much space there is for juveniles to recruit?
Yeah, so like the wild reef at Swampscott, I would say, is one of the healthier ones. So I know they're like with the fishing game data, they have been seeing recruitment and all of that. But I don't know if there's like a limitation to that. Bree, I don't know if you have anything else to add.
I would definitely say sorry. So I would definitely say that Great Bay, similar to other northeast systems, we've lost a lot of that hard substrate, so that's why, when we're building a reef, we need to add that foundation and then add live oysters on top. I think another factor that's really important is, some of our older oyster beds, when you pull up that shell, it's really brittle and can break it pretty easily, and so even some of the substrate that we do have out there isn't good quality. And that's why a lot of the efforts over at Nanny Island to sort of work down that reef is really important, because we're adding higher quality substrate out there to hopefully help to increase the recruitment.
Kelsey, those beds at Haney Island, they're not open to recreational showings.
Yeah, those are closed until 2026.
You know, during the pandemic, everyone was going out to Adams Point. And they scraped every oyster off of every rock. There's all starving people out there, I guess, but. There's other pressures, there's strange pressures on the oyster population. Any other questions?
This one's for Katie. I apologize if I missed this, but I'm wondering if you measured any like stress parameters for the oysters that were moved in the common garden. I know like Lee et al. 2020 did that in China and they found higher stress in oysters that had been moved across sites, but not necessarily lower survival. I was just wondering about that.
You mean like, certain enzymes are expressing or something
Yeah.
like that? Yeah, no, we haven't. I have lots of samples if you're interested.
Where's the funding, right? Any other questions? Oh, yeah.
Right to the chapter.
Proceed, Suba.
Katie, do you think that the oysters that died in your common garden experiment were dying because of germal infection?
Yeah, I mean, at the York River where survival was lower, there was a lot more disease pressure. There was derma, there was higher MSX. There were more pea crabs, like way more pea crab infections, than at the lower salinity site. And then we also had, the Polydora worms. I'm not sure how those were related to salinity because we haven't analyzed all of that yet, but there definitely were signs of the worms in the shells too. But I think dermo and MSX and the pea crabs definitely affected the growth and survival in the York River higher salinity site compared to the lower salinity site.
Okay, great. Any other questions? All right, thank you all.